Good afternoon. Welcome back. For the next two sessions, we're going to talk about ways to either prevent or cure a virus infection. So today, prevention is all about vaccines. Vaccines are our proven best defense against viruses, viral disease. Of course, vaccination, which we will talk about, engages the host immune system to prevent disease. And the second one is doubtful, whether it breaks the chain of transmission or not. I tend to think not. It's very hard to get that kind of evidence. But the most important function of vaccines is to prevent disease. And over time, our life expectancy, which is that graph on the right there, has increased from the 1900s. There's females and males there, two separate lines. And you can see in the 1900, on average, you could expect to live to 50 years of age. How about that? That's really young. And that has steadily increased till now. We're talking about, mid-70s, 80s. Many people go well into their 80s. And just parenthetically, in 2020, there was a dip in this because of COVID. And look at that dip in 1918. Do you know what that is? That's the influenza pandemic of 1918. Yeah, so serious pandemics have an impact on life expectancy. So we have seen this before, this graph of the immune response, antibodies and T cells on the y-axis, time on the x-axis. And that first infection there, you have an immune response. It takes some time to develop because the right cells, the right B and T cells have to be found. They have to amplify. Their B cells have to undergo somatic hypermutation and so forth, make high affinity antibodies. But eventually you get a peak of B and T cells, and then it declines. The same thing happens with vaccines. You engage the immune system. You make B and T cells. You make memory B and T cells. And eventually, antibody and T cells decline. That's the normal physiological aspect of immune responses. But there's memory in the form of B cells, memory B cells, and T cells, so that if you do encounter the virus again, you have a rapid engagement of those immune responses, the B and T cells, because you store memory. It's very quick and very much stronger than the initial response. So the result is you have a mild or even maybe an inapparent infection. So memory does not block infection. The only time immune responses block infection is in this peak here in the beginning when you have a lot of antibody or T cells. That could theoretically block infection. But the memory recall takes a couple of days to engage those B and T memory cells. So in those 2 or 3 days, you may be infected. But again, you're going to have a mild infection. Now, it's really important to understand this. All the vaccines we will talk about and all the vaccines we're not going to talk about, they do not block infection. They prevent disease. That's how they're tested. The earliest COVID vaccines were tested for preventing COVID, some kind of symptomatic disease, whether it's mild or moderate or severe. We do not test vaccines for blocking infection, and it's very hard to do that. If you think about it, you would have to vaccinate a population and then follow them for a long period of time and check them on a regular basis to see if they're infected. You'd have to do PCR or something like that. Very difficult to do and simply has not been done on a large scale. So remember that vaccines prevent disease. If you test the polio vaccine, you're testing it to prevent paralysis, not polio infection. Earliest attempts at vaccination, as we now know it, was done in a different time when nothing was known about infectious agents. In the 11th century, we've mentioned that variolation was practiced in China. That is, people who had smallpox and lived, they had pustules all over them. You would take a bit of that and give it to someone, and the ones who survived had never got smallpox again. Of course, they didn't understand that at all, and that practice spread around the world. Eventually, Edward Jenner in the UK in 1796 formalized it. He noticed that women who milked cows never got smallpox. They would get cowpox. They would get little pox on their hands from the cows, but they never got smallpox. So he said, this must be somehow protecting them. He didn't know anything about infectious diseases. He just knew that they were protected. So he took a young boy and immunized him. I shouldn't use the term immunized because that wasn't used at the time. He gave him a bit of a pustule from a cowpox on a lady's hand. And then 2 weeks later, he gave the boy smallpox in the form of a pustule from someone. He waited 2 weeks, which is perfect, right? Not like, oh, I'm not going to say that because that's an extra credit at the end of the course. I'm not going to say that, right? I was going to tell you something about contagion, but you're going to have to figure it out yourself. He waited 2 weeks, and he challenged the boy with smallpox pustules, and the boy survived. Beginning of vaccination. And we still vaccinate against smallpox, the military and other people, but it's been eradicated, and so most people do not get smallpox vaccines anymore. We used to use this bifurcated needle. We put a drop of vaccine in there, and then you scrape it into the skin. The virus replicates in the epidermis very nicely, and the scraping causes inflammation that amplifies the immune response. It acts like an adjuvant. Anyway, Pasteur in 1885 made a rabies vaccine, and he introduced the term vaccination in honor of Jenner, cowpox, vacca, Latin for vaccine, and subsequently yellow fever vaccines and influenza vaccines developed in the 30s, and then many, many more after that. And of course, as soon as Jenner's vaccine was distributed, this is a woodcut, I think, showing that if you got Jenner's vaccine, you probably would grow cow parts from your arm where you got the vaccines. The cowpox or the wonderful effects of the new inoculation. Via the publication of the Anti-Vaccine Society, already we have the first vaccine and we have an anti-vaccine society. What is it with people? What is it with people? We have since done large-scale vaccination campaigns. They can work. So on the left, top left, polio, large-scale vaccination campaigns have reduced the incidence of that disease. Measles on the lower left has vaccination in many countries has reduced the incidence, although we're having trouble eradicating because we don't vaccinate everywhere. On the right is a graph showing you the estimated measles deaths from 2000 to 2016, the top curve in the absence of vaccination. And so this is in millions. A lot of kids dying of measles. And these are on the line on the bottom is the actual measles death with vaccination. So all these lives are saved by measles vaccination. So huge, millions and millions of kids' lives saved by vaccination. And so this is ignored by people who campaign against vaccines. So today, vaccines are part of our existence. We all get them at a young age. We get multiple vaccines. We immunize kids, but also adults. We immunize our pets. We immunize our farm animals. We even immunize fish against a variety of diseases. We even immunize wild animals. To immunize animals against rabies virus, we drop bait from helicopters, and the bait has been laced with a rabies vaccine, and the animals find it, they eat it, and they get vaccinated, and it does a good job of bringing down the amount of wild rabies. So because of all this, childhood diseases are rare, and many parents say, I don't have to immunize my kids because there's no more polio or measles or mumps or rubella, which is not really the right answer, but that's what they say. They're a major part of our public health, but not of developing nations. Many countries cannot afford to vaccinate. They need our help to do so. They should have our help to do so. COVID was a great example of vaccine inequality. I personally think we should all pitch in to vaccinate everyone, but for some reason, everyone doesn't feel that way. It would not really cost a lot for us to provide vaccines to most of the world if we don't do that. Of course, there's a lot of vaccine hesitancy in many countries, here and in Europe and many others. And these are just some of the things that people say to justify not getting vaccinated. I won't go through them all, but kids should get infected naturally. Well, the point of a vaccine is to prevent you dying or having serious consequences like encephalitis. And so that doesn't make any sense. Vaccines, I'm not injecting anything into my body. There's another one said by people who usually smoke or do drugs and so forth. Anyway, these are all excuses. And when these excuses are intense, then you have a lot of problems with vaccination programs. We were lucky at being able to immunize most of the U.S. Against COVID, but we didn't get everyone because of vaccine hesitancy. And you know, the outcome of a vaccine campaign is shown by this Venn diagram. It's a matter of confidence in a vaccine. You know, we developed new vaccines for COVID, so many people didn't have confidence in them. They don't trust scientists and so forth. Complacency, thinking that infectious diseases are not a problem. And convenience, I think that's a really big one. I think in many cases, parents can't easily bring their kids to be vaccinated. When I was a kid, we got vaccines at school. We'd go to the gym, and I remember getting our polio vaccine. I think that's a good idea because they're all there. You know, mom and pop don't have to bring them anywhere. I think that would work. In this climate, though, never happening, right? Now, there can be medical exemptions to vaccines. You may have a medical condition that makes you not able to receive a vaccine for various reasons. This should be no more than 1% of the population. But in many places, it's much higher. There are some counties in Texas where it's 50% of the kids get medical exemptions, which is just not possible. It's just that the parents go to their doctors. They say, I don't want my kid to get this vaccine. So the doctor just writes an exemption without the condition for the exemption working. So those of you who become doctors one day, please do not do that. I'm asking you nicely now. It's really good for the kids to get vaccine. Now, no vaccine is 100% safe in all people, right? Because everyone is genetically different. So the mRNA vaccines we found cause a low amount of myocarditis in a certain population. You can't get around that. But the vast majority of people don't have a problem. We'll see a big problem with the polio vaccine as well. Here's an interesting graph of measles outbreaks in the US. I showed you the bar chart on the left before. These are measles cases every year in the US. And this is a vaccine-preventable disease. These outbreaks occur in populations who do not take the vaccine. And on the right are some outbreaks in the U.S. They're color-coded. The yellow are all the states with confirmed measles cases. I don't remember what year this is. This is a number of years ago. And then we have gold, cases not tied to religious community. And red, here we have cases tied to religious communities. For example, 58 cases of measles in Brooklyn in unvaccinated members of an Orthodox Jewish community where the leaders said you shouldn't get vaccinated, it's bad for you, you should get naturally infected. And then we have a Hare Krishna group here where the leaders said don't vaccinate their kids here. Another one, a pastor is critical of measles vaccination. I guarantee you this pastor did not take this virology course. And so what does the pastor know? Nothing, but probably these individuals have all spoken with anti-vaxxers who said, oh, you shouldn't vaccinate. Here's an alternative which you can buy from us. And that's really very sad that someone would put profit in front of people's lives. I just don't understand it. Anyway, you may encounter it, as I said, if you treat patients, and you have to be calm and explain it to them and ask them what their fears are and so forth, and maybe you can make some headway. So there are two kinds of vaccines. There are active and there are passive vaccines. And we're going to spend most of today talking about active vaccines. This is when you give a modified form of the pathogen or a protein from the pathogen to the person, and that induces immunity to the disease. It gives you long-term protection in the form of memory, right? Okay, active vaccines. Passive is when you put the products of the immune response into someone. Typically, it's antibodies. We don't usually do immune cells, but it theoretically could be. And this gives you short-term protection. And there's some situations where you'd want to use this. For example, that little vial there is rabies immune globulin. So it's antibodies against rabies purified from people. Actually, it's just immune globulin. The antibodies are not purified at all. But these are people who have been vaccinated against rabies. You take and you make immune globulin from them. And then if you should happen to get bitten by a rabid animal, the first thing you have to do is get injection of this antibody at the site of the bite, and it will neutralize some virus. And then, of course, you get rabies vaccination. Rabies is one of those diseases where you have enough time to get vaccinated after the bite, because it takes about 2 weeks for the virus to get into your central nervous system, and so you can immunize. So you have to do that, and then you will be protected. Of course, if you're working with animals in any capacity, you're vaccinated already. But anyway, that is an example of a passive vaccine. And of course, we get a natural passive vaccine from our mothers as we're developing in utero. This is a graph of the antibody levels during birth, during development, and after birth and into adult years in IgM, IgG, and IgA. And you can see the fraction of adult values on the y-axis. So here we have a developing fetus, and you can see the IgG in the red is going up in the fetal circulation. That's maternally passively transferred antibodies. So the mother is giving her immune experience to the child. Because the child doesn't have an immune system, it will take 6 to 9 months to develop its own. And so at birth, right here, 9 months, you have a certain level of antibody. That begins to decline slowly, about 6 to 9 months, and then it's replaced by the baby's ability to make its own antibody. Now, what is really useful here is that if you have a pandemic, you immunize the mother, you will protect the baby for the first 6 months of life, and then most of the vaccines are licensed for 6 months of age and up, at least the COVID ones. So you protect the baby for 6 months, you then vaccinate it at 6 months of age, and the baby is protected. So there's no contraindication to immunizing pregnant mothers, and you should do so in the late second to third trimester so you get maximum antibodies crossing into the fetus. We also use convalescent serum. This is prepared from blood, and this was an early result, which I really like. So this book, Fever, I told you about before, describes the discovery of Lassa virus in Nigeria in the 60s, the first emerging virus. And Penny Pinio, this lady on the right, she was an American nurse working in Nigeria, and she got infected. So they flew her back to the US, and they put her at Columbia Presbyterian Hospital, in fact, where she recovered. Not due to anything anyone ever did, but she just recovered because some people will do that. But they took some of her blood and made plasma out of it. And then sometime later, Jordi Casals on the left, a virologist at Yale, he infected himself in the laboratory with Lassa virus, and they brought him into Columbia Presbyterian. They gave him her plasma, and he recovered. He didn't die of that. And subsequently, when COVID began, we did trials for convalescent plasma. They were defective because they were given too late. So with COVID, if you give people antibodies when they're in the hospital, the viral phase is almost over and it doesn't help. So the early convalescent plasma trials didn't work. And when they were redone, given to people at the first sign of infection, they worked. So convalescent plasma is the liquid that remains when you remove cells from the blood. You prevent clotting with an anticoagulant. And that's in contrast to serum, which you let the blood clot and you centrifuge all the material out and just take the supernatant. So we take blood from people, we make plasma from it, and we give it to people intravenously. And today it's used for immunosuppressed people who cannot make their own response to vaccines. This is a very good alternative for them. At the first positive test or positive sign of infection followed by a positive test, they could get convalescent plasma. We also have used in the past monoclonal antibodies. These are antibodies against a single epitope that are directed against the spike protein. So remember, the spike protein is in the envelope of the virus. Many antibodies with these names have been made against the receptor binding domain. These block infection. Etesevimab and bamlanivimab were used extensively. They're no longer used because the variants are all resistant to them, because you only need one amino acid change in the spike to be resistant. And Omicron is diversifying so rapidly that it's no longer feasible to make monoclonals against each one of them. So this is out of our arsenal completely. Anyway, those are some examples of passive vaccines. First question, passive vaccination takes place between mother and developing fetus, involves giving antibodies or convalescent plasma to the patient, confers short-term protection against disease, although administered intravenously may still protect against respiratory disease, all of the above. Okay, what do we have? Wow, another one, 100%, good, very good. You can clap. Yeah, they're all correct. Let's talk about active vaccines now. We're going to talk about some general properties, some requirements. First, you have to induce an appropriate immune response, which, it could be Th1 versus Th2, for example, antibodies versus T cells. Often you don't know. You'd like to know what we call the correlates of protection. What correlates with protection in people? Is it antibodies or T cells or both? And if it's T cells, Th1 or Th2, antibodies or cytotoxic T lymphocytes. But you also have to show that people are protected against disease by infection. So you can't just, well, mostly you can't. There's an exception. You cannot just make a vaccine, put it in people and say, I have antibodies, and then license the vaccine. You have to show that they protect against disease. So getting a response is not enough. There is an exception, and that's a new polio vaccine that was recently developed. There isn't enough polio in the world to test the new vaccine. So they did a clinical trial, and they measured antibodies, and they determined that it was immunologically not inferior to the other vaccine. In other words, you make as much antibody as the other vaccine. On the basis of that, they licensed it because there was nothing else you could do. Some other requirements. Vaccines obviously have to be safe with minimal side effects. And remember, you can't test vaccines in millions of people. The COVID trials, the first mRNA vaccines were 40,000 people each. That's huge. You have to do the vaccine group and the placebo group. It's a lot of work to keep all that straight. So you can't do a million people. And often side effects show up when you put a vaccine in a million people. The myocarditis didn't show up in 40,000 people. It showed up when we started putting it in millions. But at least anything major that's high frequency, you would pick it up in a phase 3 clinical trial, and you wouldn't use the vaccine any longer. As I said, the vaccine has to protect against disease. Protection should be long-lasting. You don't know that until you have a few years down the road. We know now that many of the COVID vaccines induce memory that lasts three years. That's the best we can say. The WHO would like it to be cheap so they can use the vaccines in many countries, less than $1 per dose. It should be genetically stable. We'll see what that means in a bit. And storage. Do you have to freeze it? Most vaccines have to be frozen. When the Pfizer mRNA vaccine, COVID mRNA vaccine was first released, it had to be shipped on dry ice to places. That is just such a hassle. And they arranged with FedEx to do this, but eventually they got away from that and they just could ship it frozen. But this is a picture of a freezer that has been developed by WHO where it will keep vaccines frozen for a period of time. This shows you about the size of this. And these can be sent into areas without freezers. And then finally, the delivery is really important. Most vaccines are delivered by needle, and that means you need someone who knows how to use a needle and is not going to harm you. Not trivial, but oral is much easier. And then we have some approaches that may replace both of these, as we'll see. So in terms of different kinds of vaccines, we have them summarized here. We start with a virus for which there's a medical need, and that's a tough one. Well, in a pandemic, when tens of thousands of people are initially getting infected. That's a medical need, obviously. But, you know, we have some viral diseases where there are 10 cases a year in the US. Nobody's going to develop a vaccine for that. So you have to make a tough decision. Anyway, you can make a number of different kinds of vaccine. First, you can make infectious vaccines. They are given to the patient and they replicate and induce immunity, but not disease. We can inactivate infectivity. We can break up the virus and give subunits. And then we can do recombinant approaches where we clone in a plasmid, in a bacteria, or some other kind of cell, the antigen that we want to produce. And then we can, for example, produce the protein from recombinant DNA approaches. And in some cases, the protein will assemble into empty capsids, which we call virus-like particles. You can take the gene and give it as a nucleic acid vaccine. So for many years, people were testing DNA vaccines where they would inject DNA into you. Never worked. And then the mRNA vaccines were developed, and bingo, they work amazingly. So that is a brand new, really successful vaccine platform. And finally, you can put the gene for your protein into another virus vector. And we've seen a number of those made as well. So let's go through some of these. These are viral vaccines licensed in the US. There's the disease or the virus on the left, the type of vaccine, what kind of disease, or the indications for use, like where you would use it, like universal vaccination of infants as opposed to travelers going to places where Japanese encephalitis is endemic. It's not endemic in the US, so you don't need to vaccinate against it here. So many of these are given to kids. Some of them are adults only. For example, the shingles vaccine is just for adults. Yellow fever is just if you're going to places where there is yellow fever. And here's the dosage. And these are some new ones on the right that were licensed since this table from the textbook. There's an Ebola virus vaccine. And then we have the 2 mRNA vaccines for SARS-CoV-2. So these are licensed by the FDA. So that means they've gone through lots of testing. And then these vaccines have been authorized. They have what's called an emergency use authorization, an EUA. There's an Ad26 vectored spike vaccine from J&J, and then there's a Novavax spike protein vaccine. These both have EUAs. So what's an EUA? Well, when you have a pandemic, you want to get the vaccines out there quickly. You do your phase 3 trial, and you submit the data, and they give a provisional approval so it can be used. You continue to collect data, and eventually you apply for licensure, and that's how the top vaccines were licensed. So inactivated vaccines first, you take the virus and you treat it with a chemical that makes it not infectious anymore. And you can use formalin, propiolactone, non-ionic detergents, there are a whole bunch of them. And you have to make sure that you don't compromise the antigenicity, right? You don't destroy the ability of this vaccine to induce the immune response that you need to protect against disease. And I have a couple of examples to show you today of these kinds of vaccines. First is we have a polio inactivated vaccine. The disease, poliomyelitis, used to be common, as you can see in this quotation from a 1959 textbook, a common acute viral disease, no longer common in the US, of course, in many other parts of the world. And the word comes from combined roots, meaning gray marrow, inflammation of the gray marrow, basically, of the spinal cord. In the US, actually globally, polio as an epidemic disease appeared only in the early 1900s. Before that, there were just random cases here or there. And suddenly, around 1900, cases of larger and larger numbers. And you can see that big spike. Those are the baby boomers who contributed to it. After World War II, many people had kids, lots of them, and they provided more hosts for poliomyelitis, and so we had that big spike. And so all these kids getting paralyzed was a problem, and so vaccines were developed. But really the motivating factor was FDR, the president of the US, who himself had polio, I think in his 20s, and he couldn't walk. He had to use crutches and braces and a wheelchair and so forth. And he raised money with his foundation called the National Foundation for Infantile Paralysis. he encouraged kids to send dimes to the White House, and he raised $500 million, and that funded the development of the 2 polio vaccines we have today. And in hospitals everywhere, they're full of these iron lungs because sometimes kids would have paralysis of the respiratory muscles, so they'd be put in here where the chamber is filled with air, and it pushes your chest in, and then the air is taken out, and then you inhale. And this is a very gentle way to get air into the respiratory tract. Very different from putting tubes down into your lung and pushing liters and liters of air a minute, oxygen per minute, which is very damaging. But we don't have any more iron lungs, unfortunately. So one of the first vaccines developed with FDR money, if you will, was the inactivated polio vaccine, or IPV. Poliovirus treated with formalin. It's no longer infectious. In 1954, the National Foundation funded a clinical trial of this vaccine developed by Jonas Salk in 1.8 million children. It's the biggest clinical trial ever done, probably ever will be done. And it gave slightly better than 50% protection. And so it was licensed right away. These are some of the newspaper headlines. It was a big deal. People were scared of polio. They didn't want their kids to get paralyzed. But 50%. I don't know if you remember, but at the onset of the COVID pandemic, when the FDA, Peter Marks, head of viral vaccines at the FDA, that's not his real title, but he was in charge of vaccine development, said if it's better than, might not have been him also, I don't want to get emails about this, if it's better than 50%, we will license it. That's what this was, but of course it was over 90%, so that wasn't an issue anymore. But better than 50% would have been tolerated. Now, there's unfortunately an issue here called the Cutter incident, where certain batches of vaccines made by a company in California called Cutter Laboratories, they weren't completely inactivated. It was an infectious virus, so a few hundred kids got polio from the vaccine. And this began pharmaceutical company product litigation in the US. Huge lawsuits over this, and out of that came the idea that if you make a product, even if you follow the rules, if it damages someone, you're liable for it. And so in this case, the company, they broke the rules. They didn't inactivate it properly, but they were deemed liable. And those approaches last till this day. So the use of IPV in the US brought polio from 20, 30,000 cases a year down to a few thousand. But it didn't eliminate it, and we'll talk about how that was done. The way this virus vaccine works, so poliovirus is a virus that's ingested by fecally contaminated material, goes into your intestine, it reproduces in the mucosal surfaces, gets below the basement membrane, and goes into the blood and establishes a viremia. And there, if you're immunized with IPV, the antibodies will block virus infection. It will not prevent the virus from reproducing in your intestines. IPV injected in the muscle does not give you intestinal immunity. So, the US now is all IPV, and our guts are susceptible to polio. The key here, though, is only 1% of polio infections lead to paralysis. So 99% of cases are mostly asymptomatic, sometimes flu-like illness, but it is not the paralyzed kids that are spreading the disease. In fact, in the 30s, I would say, in the US, there was a practice here in New York City that during polio outbreaks, the police would go to people's houses and look for paralyzed kids and take them and bring them to the quarantine place, usually a hospital, thinking that that would stop the spread of infection. They didn't realize that there are 99% asymptomatic infections. So quarantining paralyzed kids did absolutely no good whatsoever. Another inactivated vaccine commonly used is influenza virus vaccine. Remember, this is a negative strand RNA virus with a segmented genome. There are 3 types of influenza viruses, A, B, and C. We vaccinate against only types A and types B. And the vaccine is produced mainly because we have up to 50,000 deaths per year caused by influenza virus. We make the vaccine largely in embryonated chicken eggs. It's then formalin inactivated, or chemically disrupted. It is injected intramuscularly. We make a lot of doses every year, and it's about 60% effective at preventing influenza, the disease known as influenza, in children and people under 65. Not very good in older people, unfortunately. We need better vaccines. And what protects you appears to be antibodies in your serum to the surface proteins of the virus, the HA and the NA. Some of the vaccines are just surface proteins. Yeah, the hemagglutinin by itself actually will drive the production of membrane particles with just the HA in it. And so that's actually an experimental vaccine. And then one of the licensed vaccines is just the HA protein by itself. Yeah. So they're not all made in eggs, but that was the historically first vaccine, and many companies continue to do that. As you can see, some are now produced in cell culture of various sorts and not all in eggs. But they're all not as good as they could be. Now, the problem with influenza virus is that the surface proteins change often every year. So in the first months, at least in the northern hemisphere, in the first months of the year, we have to pick a new vaccine for the fall season. So starting in January, we have to decide what strains are going to be in the vaccine for the fall. And so there's a lot of flu surveillance globally. As I told you, they pick what's thought to be the new strain, and it's a hit or miss proposition. And then they take the HA and the NA of that strain and they put it into another influenza virus that grows really well in eggs, and that's the vaccine for the year. So the current year, 2022-23 season, which is almost over, it had two different influenza A components, an H1N1 and an H3N2, and two different influenza B components. So it's a quadrivalent vaccine. It has 4 different kinds of influenza viruses in it. Is there a maximum number of antigens that you could put in? So yes, because the more you put in, the less of each, because you can only give so much total antigen at once. And I was talking to someone who was making mRNA vaccines for all the different influenza viruses combined, right? Like 15 or so or 20. And we asked him that. And he said, yeah, beyond a certain number, you get diminishing returns, and you may want to do it in stages. Yeah. But of course, there are many bacterial antigens, vaccines that have many different antigens. Pneumococcal vaccines and so forth have many, up to 20, and it works reasonably well. This is the process for selecting an influenza vaccine every year. So there's year-round global surveillance. So GISN is the Global Influenza Surveillance Network, which has been put together by the WHO. You know, the first 2 months of the year, you pick the strains, you make the reassortants with the good viruses that grow in eggs, you standardize the antigen, you standardize the potency, and then you formulate and package, and then you start to vaccinate at the end of August for that full season. And sometimes you hit the right combination, and sometimes you miss, and there's more disease than you would like. The problem, as I said, is that the virus changes, and particularly the hemagglutinin protein undergoes what we call antigenic drift. A single amino acid changes every so often, and these are the epitopes on the influenza hemagglutinin where antibodies bind, where monoclonal antibodies bind. And the ones at the top, that's the tip of the HA that binds the sialic acid receptor. Those change quite often, and often one amino acid change in any of those epitopes is enough to need reformulation of the vaccine. The epitopes on the stalk here, those are conserved, and people are trying to make vaccines that are directed against them because they don't change nearly as much. And so we're going to call those universal influenza vaccines, because in theory, maybe you would not have to immunize people every year. Even every 10 years would be good. Question 2, which statement about inactivated viral vaccines is incorrect? A, chemicals can be used to inactivate infectivity. B, they do not replicate. C, they can be dangerous if inactivation is not complete. D, antigenic variation can make them ineffective. E, none of the above are correct or incorrect. All right, what is the answer? So none of the above are incorrect. So the chemicals can be used to inactivate infectivity for sure. They don't replicate. They're inactivated. They can be dangerous if inactivation is not complete, like the Cutter incident. And antigenic variation can make them ineffective, as we saw for influenza virus. Excuse me. Okay, let's talk about subunit vaccines. There are two approaches to subunit vaccines. First, we can take the virus and just break it up chemically into subunits and purify them. And some of the flu vaccines are like that. But the modern way to do it is to take the gene that you're interested in. If you're interested in the hemagglutinin of influenza virus, you put it in a plasmid, you put it in some kind of plasmid vector, and express the gene in bacteria or yeast or insect cells, you get the protein, and you immunize with the protein. And sometimes, if it's a capsid protein, it will assemble into empty capsids without any genome in it. They're called virus-like particles. And we're going to look at some examples of this. First one is the shingles vaccine, Shingrix. So varicella zoster virus is a herpes virus, which when acquired as in childhood, causes chicken pox. And before we immunized kids against chicken pox, they would get chicken pox, and then the virus goes latent in you. And then at age 50 or up, it would reactivate, and you would get shingles, which are these bands of vesicles along a specific dermatome, a specific part of the skin innervated by certain ganglia. So a vaccine was made for people who got chickenpox before the vaccine to prevent shingles, and this was what it's called, Shingrix. It is one of the glycoproteins of varicella zoster virus. It has a lot of glycoproteins. The particular one is glycoprotein E. It's produced in mammalian cells, actually hamster cells, CHO. It's secreted. It's then mixed with an adjuvant and injected. And this is a really good vaccine. It's very good at preventing shingles. I just got it actually last year, and now I should never get shingles again. We'll see. I'll let you know. Then we have another subunit vaccine against hepatitis B virus. And this is a cancer vaccine. This prevents cancer caused by hepatitis B virus. It does not prevent infection. None of these are preventing infection. But this one will stop virus replication to the extent that it no longer causes cancer. So remember, hepatitis B virus is associated with liver cancer. And so this vaccine was developed to try and prevent that. It is the surface antigen of the virus. So remember, the virus is made up of a icosahedral nucleocapsid with a very weird genome, a gapped genome made by reverse transcription. And it's enveloped, and the envelope is the surface antigen of hepatitis B virus. So the gene for that was cloned into a vector and expressed in yeast. And the protein, when expressed, assembles into empty particles of different sizes, as you can see here. So there's no nucleic acid in them. They're mixed with an adjuvant and then injected into you and provides very good protection against hepatitis infection. Yes?
What's an adjuvant?
Okay, I was wondering if someone would ask. I'm going to explain it in a minute. It's a chemical that you mix with the vaccine to improve immune responses. But we're going to go into it in a bit of detail. Another subunit vaccine, human papillomavirus. It's another one where you produce a single viral protein. It assembles into an empty capsid. So papillomaviruses, right? These are these DNA viruses, double-stranded DNA, circular, small. They can cause warts. There are over 170 different types. And so if you ever had warts on your hands or on your feet, if you walk around at a health club, you go take a shower and you walk around barefoot, you're going to pick up papillomaviruses on the bottom of your feet because someone else is shedding them, and you're going to get warts. So you really should wear flip-flops in the showers, okay? Because you're going to get warts, I guarantee it. Anyway, there are a lot of different types, but some of them are sexually transmitted. They're not just picked up on the bottom of your feet. It's the most common sexually transmitted disease in the U.S. On the right is the bar graph showing the incidence or the percent of people, men and women, all Americans, Black, Hispanic, white, and Asian, who are seropositive for these papillomaviruses that are sexually transmitted. So quite a few people. Now, some of them cause low-risk genital warts, okay? And they do not progress to cancer. But other types, certain specific types, including type 16 and 18, cause a variety of cancers: cervical, vaginal, penile, anal, oropharyngeal. About 31,000 cases of these cancers every year in the US, and several thousands of deaths from these cancers. And so these genital types, 18 through 59, basically nearly half of Americans, you can see, are infected with them. So it was decided a long time ago to develop vaccines against these. And the vaccines are made by a couple of different companies. And they're basically the viral capsid protein produced in either yeast or in insect cells. So the capsid has one major protein. Its gene is cloned into a vector, and in this case, we're producing the protein in yeast or baculovirus insect cells. They assemble into empty capsids. The capsids are mixed with an adjuvant, and they're injected intramuscularly. Now, these vaccines should be given to kids before they become sexually active. Otherwise, you're already likely to be infected, and the vaccine's not going to do any good. Likewise, it doesn't help to take them after you're 45 because there are not enough years left in your life to develop the cancers, basically, to provide the protection. But in the right age group, these are great at almost 100% protection against these kinds of cancers. So really, really effective vaccines. They do not prevent infection. They just prevent the cancers at a very good rate. So many parents don't want to immunize their kids because they think it'll make them promiscuous. And many studies have been done showing that that's not the case. I used to go to high schools and talk about, I was invited by an English teacher to talk about the Henrietta Lacks books. And so she wanted me to explain the science. So I would explain it, and I talked about this vaccine. And every year someone would say, why do my parents not want me to get this vaccine? And I said, because they think you're going to become more sexually active. And they always laugh. So these are some ideas for future influenza vaccines. One is to make the HA protein in plants. You can make the HA protein in cells, and what it does is it causes the membrane to bud out, and you get these vesicles with HA protein in them. But if you do it in plants, it's incredibly cheap. A square meter of plants will make 20,000 doses of vaccine at less than 20 cents a dose. So you can put the HA gene into plants, and you just crush the leaves and you harvest these particles. And they're in clinical trials now to see if they work well. Now, these subunit vaccines have some advantages, right? Recombinant DNA, you can make them really quickly. There's no viral genome, there's no infectious virus, so they're very safe. But they tend to be expensive. The HPV vaccine, the course, 3 doses, I think, is $150. They have to be injected. And unless you put adjuvant in them, they have poor antigenicity. So why is this that they have poor antigenicity? Well, proteins don't replicate or infect. They cause poor inflammation on their own. And remember, good inflammation, Calor, Dolor, Tumor, Rubor, is needed for a good adaptive response. And so what we do is we add adjuvants to make up for that. So an adjuvant is a chemical or a compound that we add to these non-replicating vaccines to improve their immunogenicity. Basically, the adjuvant is causing inflammation on its own. And the only vaccine we don't do this with is poliovirus, inactivated polio vaccine. For some reason, it doesn't need an adjuvant. So adjuvants act by causing inflammation. They also tend to keep the antigen at the injection site and have it released more slowly, which helps apparently immune responses. And these are some adjuvants that have been licensed. So the hepatitis B vaccine has aluminum hydroxide or aluminum phosphate. The Shingrix has an adjuvant called AS01. It's monophospholipid A, which is shown there no, so it's not shown there, lipid A, which is a ligand for toll-like receptor 4. So basically, it tickles toll-like receptor 4, which then signals and makes cytokines, and you get inflammation. AS4 is in Cervarix, one of the papillomavirus vaccines. This is actually lipid A, which is part of bacterial lipopolysaccharide, which is shown right here. So LPS is highly stimulatory of innate immune responses, so it causes inflammation. And then there's one, MF59, which is an oil and water emulsion that is mainly used in Europe, and that's what it looks like there. I didn't tell you, actually, these papillomavirus vaccines, you can see Merck makes 2 different kinds with multiple serotypes in, and so they made initially 6, 11, 16, and 18, and then it was clear that other serotypes were also involved in cancer, so they included additional serotypes, and there may even be more than this now, actually. Another technology I like, 2 technologies. First, on the left there is the microneedle patch. So it's a small plastic patch with tiny needles in it, and you would saturate it with vaccine and then put it on your shoulder with a Band-Aid. And the needles go into the epidermis, which is a really good place to deliver antigen, a lot of antigen-presenting cells there, and it slowly diffuses in. There's no needle needed, so anybody could put this on. I think this is going to revolutionize vaccination in the next 10 years. It's going to replace needles, in my opinion, and I think that will help get more people vaccinated. The other issue is that you have to freeze many vaccines, and it turns out that if you freeze dry vaccines in sugars, it thermostabilizes them. So there's an experiment on the right where we're looking at the infectivity of the vaccine in PFU over time at 2 different temperatures. So this is influenza A vaccine at 40 degrees Celsius. You can see in 2 days, all the infectivity is gone. But if you freeze it if you lyophilize it with sugars, the sugars stabilize the infectivity to a certain extent. So people are working on this to try and get it better. And so something so simple as this may be a way to get rid of having to freeze vaccines, which is an issue in many countries, as you might guess. Here's an approach to a universal influenza vaccine to try and trick the immune system to making antibodies against the stem region epitopes, which don't vary all that much compared to the head region epitopes. The problem is that if you inject the HA into someone, the head is immunodominant. So most of the antibodies are made against the head. So what do? You have to trick it. So here's the one approach. You make chimeric hemagglutinins. So say you want to immunize against an H1 stem. So you make a chimeric hemagglutinin with an H9 head and an H1 stem. The H9 is not found in people, it's irrelevant. Then you wait for the antibodies to develop. Then you boost them with an H8-1 chimera. So now you have a different head. So the immune system is not going to make a memory response to the head. It's going to see a new head, but it will make a memory response to the stem, and then you do it a 3rd time with a different head and an H1 stem. And so the idea here is that you're making the stem immunodominant by this procedure, and these are being tested to see if, in fact, this works. But there are other approaches that are being used as well to try and make universal flu vaccines. In other words, the goal is, can we make a flu vaccine that doesn't matter if the virus changes every year in that head, in that globular head? Is that possible? Maybe you could have it once in your lifetime, or maybe you could have it once every 10 years. That would even be better than having it every year. All right, question 3. What are some requirements for an effective vaccine? Low cost, ease of administration, provides long-lasting immunity, minimal side effects, all of the above. Okay, what did we do? Another 100. All right, you guys like vaccines, don't you? So that's 5. I think that's a record, of all the years here at Columbia. What do you think about that? You're not impressed, huh? Okay. Finally, we're going to talk about what I call replication-competent attenuated vaccines. Infectious vaccines. Some people call these live attenuated vaccines. I don't really like it because virus particles are not living, right? So I call it replication competent attenuated, but you will see live in the names, I'm sure. And here, the virus is replicating. The vaccine virus is replicating. It stimulates an immune response, but it doesn't cause severe disease. It doesn't paralyze you, doesn't cause severe flu, and so forth. So mild or inapparent disease. And the idea is when you have an inactivated virus vaccine, you typically have to give multiple doses to get a good immune response because the virus is not reproducing. And you can only give so much antigen, so you give multiple doses. And we've decided that COVID vaccines are good at 3 doses. Many other vaccines are 3-dose vaccines. But the attenuated infectious vaccines on the bottom are self-amplifying. They're replicating in you, and so you presumably don't need as many doses. So measles, mumps, rubella vaccine is typically 1 dose, although some people are thinking of making it 2 doses now. All right, so how do you make an attenuated virus vaccine? There are 2 general approaches. One is the empirical approach, where you just take your virus and find some conditions where you can grow it to make it not cause disease. And so here's an example of that. You take a pathogenic human virus, and then you grow it up in cells in culture, and then you passage it, say, in monkey cells. And you do this many, many times until now you isolate a virus that grows really well in monkey cells, but maybe doesn't cause disease in humans anymore, which you'd have to test by a clinical trial. And so an example of this, another example would be the yellow fever vaccine, which was made in the 1940s by passaging the human virus hundreds of times in I think it was chick embryo, or chick embryos, or chick embryo cultures, I don't remember, but hundreds and hundreds of times until it lost its pathogenic properties. So FluMist is a flu vaccine that is an infectious attenuated vaccine. It's given intranasally. You can see this little girl is having it sprayed into her nose. It's a syringe without a needle on the end, right? And it goes up into your nasal passages and reproduces. And it is made by, so there's a master donor strain, which is cold adapted, temperature sensitive, and attenuated when tested in ferrets, which is a model for influenza. So cold adapted means it prefers to reproduce in the upper tract, which has a lower temperature than your lungs. Temperature sensitive means it will not reproduce at the higher temperatures of the lungs. And then attenuated, it doesn't cause disease in ferrets. And so every year when you need to change the strain, you can take the HA and the NA of the prevailing isolate that's circulating and put them into this master donor that has all these properties. It just reproduces in your nasopharynx. So this is a great idea, but it's no better than the intramuscularly injected vaccines that protection against severe disease. I mean, they do protect you reasonably well against severe influenza, but this is no better than the intramuscular vaccines. It doesn't give any longer protection. It should, right? Because it's a natural route of infection. So something is missing, and maybe, I'm not sure it will ever be worked out, because I think people are really focused on mRNA vaccines now, and that's all that's going to be developed. There are also attenuated poliovirus vaccines. This is the Sabin oral polio vaccine. So Sabin was the virologist who developed this. This virus you drink. So this, when I went to get vaccinated in my high school gym, I remember there was a little sugar cube there in the cup. It was red, and they just let us eat it. And that had this vaccine in it. And it goes into your intestine, it reproduces. You shed it for up to a month. It protects your gut for a certain period of time, but also will protect your blood in case the virus gets in. And so we switched to OPV in 1962 because IPV wasn't eradicating polio, and we said, this is going to be better. And in fact, we eradicated polio from the US in 1979. And so Albert Sabin made these viruses empirically, as I said, by passaging. There are 3 serotypes of polio. He passaged them in different cells in culture, and he, in some cases in non-human primates. And at each passage, he would test it in an animal and say, is this no longer causing paralysis? And then he finally got candidates of all 3, tested them, and they were licensed in 1961. Now, in the 80s, when we got the ability to do genome sequencing, we figured out where the mutations were in these oral poliovirus vaccines. And you can see the 3 serotypes, and there are very few changes throughout these viral genomes that are responsible for eliminating the ability to cause paralysis. You see they all have a change in the 5' non-coding region in a similar area. And in fact, if you look at the viral genome, remember the 5' end is highly structured. This particular stem loop 5, that's where all 3 serotypes have single base changes that are responsible for the majority of attenuation, that is the inability to cause paralysis. All right, so that's very interesting. The problem is that these changes are genetically unstable. So here's an experiment where a virologist in the UK, he and his wife had a kid. They gave it polio vaccine, and then they took all the kids' diapers and brought them to the lab and sequenced the virus that the kid was excreting. So the vaccine, here we're just looking at base 472, which is one of those that's really important for attenuation of the type 3 vaccine. The vaccine has a U. After 24 hours, David is excreting U-containing virus, 31 hours U, 35 hours, boom, he's starting to revert to C, and now after 2 days, he's shedding fully virulent polioviruses, which if you stick in a monkey, causes a lot of neuronal damage. So that's the wild type base that Albert, he replaced it with U, but he didn't know what he was doing. He was just passaging these viruses in different ways. So this happens in everybody who gets polio vaccine. But not everyone gets paralyzed, fortunately. Now, when we started using this vaccine in the US in the 60s, it eventually brought down the wild polio. So this line is all the total paralytic polio cases. You can see it brought it down to a low level, but not 0, because these bars are cases of polio caused by the vaccine. So in 1.4 million doses given to kids, they get paralyzed. And that's 10 to 12 cases a year. So in 2000, it was decided we cannot do this anymore. This is no longer a good public health strategy. So we switched back to IPV, no more vaccine-associated polio, okay? So this is the one vaccine that I think is questionable, because I know this is rare, 1 case in 1.4 million, but if it's your kid, it's not really good, right? So I'm not really sure why we did this. Now, this is significant because back in '88, the WHO said we're going to eradicate polio, and eventually we're going to stop vaccinating by 2010, which is past. Where are we? Well, first of all, can you eradicate a viral disease? Only one human viral disease has been eradicated, and that's smallpox. There are actually 3 features essential for eradication. I've changed my mind since last year. I'm going to update this slide right now in real time. Okay, the 3 features you need. Replication only in one host. Number 2, vaccination induces lifelong immunity. And number 3, asymptomatic infections do not contribute to spread. Really important. Because if you have asymptomatic infections, you don't know who has smallpox or any viral disease. The issue with smallpox is, okay, it only replicates in people. Vaccination induces lifelong immunity. And everyone who's infected gets pox. So you can tell where the virus is. Wherever there's virus in a person, they have pox. It's not the case with polio, right? 1% of polio infections cause polio. So 99% are asymptomatic. So how do you know where they are? So far, all they do is look for kids with polio, go in and vaccinate extensively. So I think this means we can't eradicate polio. Anyway, we started this campaign in '88, 71 polio-free countries. 10 years later, 156. 20 years later, 191 polio-free countries, doing really well. This is funded by Rotary International, the Gates Foundation, and others. So today, we have a few hundred cases of polio in the last 12 months. Types 2 and 3 have been eradicated. Wild types 2 gone from the planet, as far as we can tell. The only thing left is type 1, which is causing outbreaks in Afghanistan and Pakistan because we can't immunize. The Taliban prevents the immunizers from getting in. They shoot them, so nobody wants to do it. And now it's spread to Africa from these countries. So in Africa, the problem is it's hard to maintain high levels of vaccination coverage because the countries have all kinds of issues and vaccinators have trouble covering the kids, et cetera. So we have type 1 there. But the orange dots are outbreaks of vaccine-derived type 1, and the green are outbreaks of vaccine-derived type 2. So wild type 2 is eradicated, but vaccine-derived type 2, remember, it's reverted, it's circulating, and whenever coverage of the vaccine drops, you have an outbreak of polio. So not only is the vaccine causing polio in 1 and 1.4 million kids, but it's circulating and causing more polio outbreaks. And what do they do when there's an outbreak? They go in with OPV. In fact, here, this graph I showed for 15 years, it never had the Western Hemisphere in it. But now it's in it because there was one case of polio last summer in New York, right there in Rockland County, down over in New York State on the other side of the Hudson River. Yes?
Didn't they also detect it in the wastewater?
Yes, I'm getting to that. I'm getting to the wastewater, yes. So the problem is if vaccination coverage drops, these vaccine-derived viruses will cause polio. And remember, in the US, we use IPV. So the virus is probably circulating. The OPV is probably circulating. We don't know it because before last year, we didn't check sewage to see if people were excreting the virus. So one paralytic case caused by vaccine-derived Sabin2. We found viral genomes in local wastewater. It's a recombinant of OPV2 and Coxsackie virus. They're probably all throughout the US, and that's because we don't, IPV does not protect the gut from infection. This Rockland County case was in an area where vaccination coverage was, here's by age percent. By age 2 years, you can see these counties 0 to 50% vaccination coverage, and that's where this case was. So the solution, of course, is to vaccinate, and then you don't have any polio. And right now, CDC is looking in the rest of the US to see if there's polio in wastewater. Wastewater is not going to spread polio to you, but it's a sentinel. It tells you that there are people who are in the area who are infected and shedding fecal material that's going into the wastewater. Now, a couple of years ago, scientists said, can we make an OPV that doesn't revert, that doesn't cause paralysis? So they introduced changes in the genome. They introduced changes in the UTR that would prevent reversion, and they introduced a change in the RNA polymerase to reduce the error rate. They introduced a change to reduce recombination in the polymerase. This CRE element was moved to the 5' UTR to reduce recombination. These viruses, they're called NOPV2. There's less reversion in mice, and they're immunologically non-inferior, as I told you before. So they were introduced, they were authorized by WHO in 2020, and so far they've been given to 600 million children. The 5' end doesn't revert, but just recently, 7 kids were paralyzed after being given NOPV2 in Burundi and DRC. There's also virus in the environment. This is an improvement over OPV because the same number of doses would have caused 30 to 40 cases of paralysis with OPV2. So this is better, but it's not perfect. You haven't prevented this reacquisition of neurovirulence. And I'm just worried about the NOPV2 in the environment. I'm afraid it's going to circulate and cause cases of paralysis whenever vaccination drops. So we'll have to see what happens there. But remember, if you eradicate a virus, you could always make it again. If you have the genome sequence, you could make any virus. So here's an article from PLOS One, construction of an infectious horsepox from chemically synthesized DNA fragments. This virus was eradicated or extinct. They had the genome sequence. They synthesized the genome, put it into cells, and out came virus. So you could do this with any virus. So eradication is only as good as your surveillance. So now we have a new class of vaccines, mRNA vaccines made by Moderna and Pfizer. You take a DNA encoding the spike protein. You have a promoter for an RNA polymerase that will make a large amount of mRNAs in vitro. This is packaged into lipid nanoparticles, and then these are injected into your muscle. Now, the spike that's put in to these mRNA vaccines have two prolines added C-terminal to the S2 cleavage site. So when the spike binds to ACE2 and then it's cleaved at the cell surface by, say, TMPRSS2, the spike undergoes this conformational change to trigger fusion. And that form, the post-fusion form, doesn't have the right epitopes to induce monoclonals or antibodies that would protect you from infection. But the introduction of two prolines here prevents this conformational change. It maintains the spike in the pre-fusion form, which has all of the important epitopes. So this is injected intramuscularly. Your antigen-presenting cells will take up the lipid nanoparticles. The RNA will be released in the APC. The APC goes into the lymph node. It will process the protein, present it to T cells, and then the T cells will become activated in the scheme that we have talked about already. So that's how this works. And as you know, they are very, very good at preventing COVID. The problem is we don't give COVID vaccines, of which there are others as well, of course. This is coverage globally, total doses per 100 people. The light colors are not enough. So you can see much of Africa is undercovered. And then there's a lot of countries where we don't have information for them. And we will talk more about the COVID vaccines and some of the issues with them later when we talk about the pandemic itself. And so next time we will talk about the other way we can deal with virus infections after they've already begun using antivirals.